So where it is, Bharat, this your company in Bangalore?
Hello.
Hello, Bharat.
Yes, sir. Our corporate office is located at MG Road, Bangalore.
Oh, yeah. Okay,
Yeah.
okay. So you are working on, I mean, the company is actually working on bioinformatics?
We are working on plant-based molecules, sir.
Molecule, okay.
Yeah. We have Dr. Rishikesh Damle, our MD.
Okay.
Welcome to you, sir.
Hold on. I'm stuck in a very bad traffic.
Oh,
I think even my face is not visible there in that. Okay, you see it. Hi, doctor. How are you?
I'm good.
Yeah. So I'm in the car. So there is, I think, some accident, and we are stuck for past 1 hour. So hopefully we will reach before you end your, talk.
Oh, okay.
So and my connectivity is also poor on this flyover. So I will switch off the video and I will. Bharat. Dr. Bharat.
Yeah.
Bharat, you have mute.
hello. 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 Hello.
Yeah. Hello, Bharat. Hello, Bharat.
Yes, sir.
Hear
Yeah.
you.
Okay.
Bharat, can you hear me now?
Hello.
Bharat, you can start by 7:35.
Okay, sir. Sure. Not
And
a problem.
Manoj is not there. From AMAI, somebody is logged in.
Okay.
We will, I think they will do the welcome speech.
Sure. Sure. Not a problem.
Yes, sir. I have joined in the place of Manoj Kalu for the welcome speech.
Dr. Anand, sir.
Yeah.
Yeah, welcome. Yeah.
Okay.
Thanks.
Okay, sir.
Hello everyone. Good evening everyone here, and today we are on to an hour with an expert lecture series 5. Today we have Dr. Bharat Rajguru from Department of Biotechnology, MIT Manipal, and we also have Dr. Shivan Ganju, Chairman, Atomid Pharmaceuticals Private Limited, with us as a panel member. We have Dr. Rishikesh Damle, MD and CEO, Atomid Pharmaceuticals Private Limited, Bangalore. And we have Dr. Lata Damle, CSO, Atomid Pharmaceuticals, Bangalore. We have Dr. Anand S. as a panel member from Government Ayurveda College, Trivandrum. And we also have Manoj Kalur from AMI and AVVS. And today we are going to have a privilege of listening to our expert, Dr. Bharat Rajguru. He has completed his Bachelor of Engineering in Chemical Engineering from National Institute of Technology. And later on, he has continued his MTech studies in Chemical Engineering in Indian Institute of Technology, Bombay. Further, he has enrolled for PhD in Chemical Engineering at Wane State University, USA. Further, Dr. Bharat Rajguru has joined University of Minnesota, USA as postdoctoral fellow. So after completion of his postdoctoral studies, he has joined Department of Biomedical Engineering, Case Western Reserve University, USA as research associate. Dr. Bharat Rajguru is having active collaborations with IIT Madras and KMC Manipal. And Dr. Bharat Rajguru has a US patent granted for his research on dendrimers for sustained release of compounds. And he also owns a Indian patent on system and methods for synthesizing isoniazid with hydrophobic moieties and encapsulating biodegradable polymers. Dr. Bharat Rajguru has also published more than 15 research articles in peer-reviewed international journals with high impact factors. To name a few, he has published his research findings in the Journal of Biomaterials with an impact factor of 8.38, Nanomedicine with an impact factor of 6.2, molecular pharmaceutics with an impact factor of 4.35. Fortunately, I am also a co-author in his one of the research articles, and Dr. Bharat Raj is also an author of book chapter on optimization of glucocorticoid encapsulated PLGA nanoparticles for inflammatory diseases, published by Springjo, Singapore. Dr. Bharat Rajguru is a recipient of research grants from VGST, BIRAC, and Manipal McGill Center for Infectious Diseases for his research on nanomedicine. Dr. Bharat Rajguru has shared his ideas in the different conferences and symposium as an invited speaker. Today, it's a great privilege for us to listen to Dr. Bharat Rajguru. Over to you, sir.
Thank you. Thank you, Bharat.
There is a welcome speech by Anand. So Anandji, please welcome before our guest starts.
Okay. Utmost respect to the all dignitaries. A very warm good evening. And Dr. Shiban Ranju, the chairman of Atrimed Pharma, Dr. Bharat Rajaguru, who is taking the session today, Dr. Krishikesh Damle, CEO Atrimed, Dr. Sadat Dinaga, General Secretary of Ayurveda Medical Association of India, Dr. Manoj Kalur, Arya Vaidya Vilasini Vaidishala, Dr. Uday Kumar sir, Dr. Lada Damle, Dr. Shaiju, all the dignitaries across the globe and eminent personalities, dear doctors and dear experts. Today, in this series of an hour with an expert, we have a great session with the subject application of nanomedicine to treat some of the difficult diseases to cure. This is a very promising and very sought after area, since in the field of medicine, the nanomedicines are very much getting relevance today. And especially in this scenario of this infectious disease and the disease of devastating disease like cancer, we have greater role in nanomedicine. And even characterization of single nanomaterial to nanorobotics, there is a large area before this technology. And I'm very happy to be have a session with this great expert, and I'm sure that in the coming days we have much collaborative works to open in multidisciplinary regard, since in many of the basmas in Ayurveda, Rasashastra branch in Ayurveda, in some of the basmas, the metallo-mineral compounds, we have identified some nanoparticles, even ranging to 2 nanometer. Even it is much small than a cell. So it's very interesting for all of us to hear the invited speaker, and I welcome in name of Ayurveda Medical Association of India, Atrimed Pharma and Arya Vaidya Vilasini Vaidishala, and all present here to welcome this eminent speaker to have a great session here. And I am sure that it will open up in some discussion, and it will lead to multidisciplinary approach. And we have to dig out the treasures of our traditional wisdom, Ayurveda, the wisdom of this country, in connecting with this emerging field of nanotechnology. So I warmly welcome the speaker, Dr. Bharat Rajguru, and all the eminent personalities here, and those who are watching across the globe to this session. Thank you.
Thank you, Dr. Anand. Shall I take over now?
Okay, sir.
Yes.
Okay. Good evening, everybody. Today, whatever my experience with the nanomedicine, I'll speak, and then how it will be useful to cure some of the very difficult diseases which are considered non-curable in some cases. So first, I'll start with eye. Why eye is so important is, just see, this is the actually eye. This is the anterior part of the eye, and this is the posterior part of the eye. And the eye anterior part and posterior part is separated by something called vitreous chamber. It has a 99% of water, and then the main goal of this is to keep the posterior part in an aseptic condition. And because the posterior part of the eye connected to the central nervous system, so it is very an evolution how we evolved. It is since it is a central connected to the central nervous system, it is highly protected. So the disease is something which occur in the posterior part of the eye, very difficult to cure. One of the reason is, though you know the drug will not reach to the posterior part of the eye, because suppose you put anything eye drop or something, it has to diffuse into the anterior chamber and then to the vitreous chamber, and then it has to go to the posterior part. Probably around 2 to 3% of the drug gets in. And since posterior part is connected to the central nervous system, the immune system is highly active in the posterior part of the eye. Something happens, a small cell death will trigger an immune response, high immune response. In the process, neuroinflammation is, one of the things which occurs drastically, and that will create a major problem. Okay, so then how to solve this problem? So some of the diseases which are all age-related macular degeneration, glaucoma, retinitis pigmentosa, and diabetic retinopathy. These are the diseases which is happen in the posterior part of the eye. Glaucoma is because of the neuroinflammation. The swelling of the eye occurs. So that is a glaucoma. And then say there is very few data is available about India. So I could say that when around 200,000 new cases are going to come up only in USA every year, which is on age-related macular degeneration. So I tried to find out the data in India, which we really do not have the data. So you just see that as the aged population increases, then these diseases, ought to go in the society. So that's why we have to give much importance how to cure or keep it in a low key. Why curing is difficult? I'll come to that and later. So as I said, this is the photoreceptor cells are the retinal cells which I have put here. So it translates light image into electrical signals. So that's why it is connected to the optic nerves, and this is connected to the central nervous system. So degeneration of these photoreceptor cells that leads to blindness. So that is the retinal cells. So this is some drug and then also it has a retinal blood barrier. So obviously there are barriers which is in a highly protected region. These barriers makes it very difficult the other foreign particles to go there. Because an evolution, what we have evolved, this is a highly protected region. So one of them is a blood-brain barrier. So whatever it will not go to the brain easily. So there is a barrier. Similarly, similar to blood-brain barrier, there is a retinal blood barrier. So something which is the blood getting into retina, there is a barrier for that, and whatever it is in the blood, it will not get into the retina. So most of the drugs which are all xenobiotic are like which are not produced in the body. Then in the process, it may not allow it to pass through the retinal blood barrier. So that is again one of the main problem. Then how these diseases are right now are treated. I would say up to 90 there was nothing called a curing. People used to put, say, that most of the time it is a steroid has to be put. They used to put on the as an anterior part. They put in an eye drop and it's supposed to reach in the diffuse here, and reach here. Then afterwards they realize, it is very difficult and the therapeutic value will not reach there. Then they modified, and then can we inject something here into vitreous chamber? And as I said, vitreous chamber is to protect the posterior part. Vitreous chamber is present to maintain the aseptic condition and also to maintain the pressure of the eye. And then say when you inject, you cannot inject every day. So you have to inject, and then highly specialized surgeon should inject into vitreous chamber. And then, probably 1 month or 2 months once, you can inject again one more dose, or as you avoid, say 1 year or 2 years once if you inject, then, it would be a great thing. So that's what people have tried. One of them is, say, the non-biodegradable polymer device, which releases drug up to 30 months, almost a 0.2 micrograms per a day release to keep the glaucoma. It's a fluocinolone acetonide. It's a corticosteroid. It releases of around 30 months. That's what the things. But you just see that it is a non-biodegradable implant. So that since though it is a biocompatible, the polymer is a biocompatible with the matrix, it releases slowly, but it will not degrade. So after 30 months, it floats around in the vitreous chamber. Then again, one more dose. Assume like somebody at the 50 or 50, 55 years has to take this, and then he lives up to 75 or 80 years. Then so many non-biodegradable devices will float around in the vitreous chamber. That may not be the proper idea. Then what people have also something called a scleral plug. In the sclera, they put a plug of a polymer which releases drug slowly. And then, it can be taken out. The plug can be taken out. So this was also, people have tried. And then, it has to get here and then release. But though not many are in the market right now, but people are working in the phase 2, phase 3 trial. And then say, what about the nanoparticles? If you put the nanoparticle here, what happens? So that is one of the thing. So if you put the nanoparticle here, the people have shown, or the researchers have shown, it is taken up in the posterior part of the eye, say around 200 to 300 nanometer, and gets into the retina and stays there for 4 months. So that is a very good thing. So that is the, say, PLA nanoparticle. This is the seminary paper which is published in 2003 or sometime. So then, it gets in there and releases slowly. It stays there for 4 months. And then say if it is a microparticle or an implant that is there in the market right now, a corticosteroid, dexamethasone, is they put in here and it releases, floats around in the middle and releases drug. That is almost like a implant, and it is a biodegradable implant. The biomaterial degrades. It is a biocompatible, and it degrades slowly and metabolizes in the body. So that is a FDA approved. It is there, and though it is not there in India, it is there in US right now. It releases drug, and then from here it goes there. So now whatever my research is, something a 5 to 6 nanometer a polymer, if we get in here, does it get into the retinal cells and release drug slowly? Okay, that is and release drug slowly for say 1 month. If I inject only one injection, can I do that? So we selected something called a dendrimer. So this is a dendrimer. Dendrimer is a tree-like structure. You make a polymer. Through polymer, you make a dendritic structure. Dendron in Greece means it's a tree, and then you can make whatever size you want, and then you can create the outer surface with the whatever uh, 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 group, or like a group which is a OH, NH2 or COOH group. These are the groups usually which are used for conjugation. So whatever groups can we make? So this can vary, and then molecular weight can be varied, and the size can also be varied. So what we selected is we made a polymer which is a polyamidoamine polymer. So polyamidoamine is ethylene diamine is the major polymer with a dendritic structure which it makes. Say, as I mentioned here, say that red is a generation of zero generation. So initially you make a polymer zero generation, and then you add again, say, green generation one, and then say again you polymer. As the branches increases, generation increases. So one of the important thing is, as the generation increases, obviously the molecular weight of the polymer also going to increase. So we stopped it around the generation 4. The generation 4 with the OH group. See, NH2 group is positively charged. Since the cell membrane is negatively charged, then there is a chances that the more and more polymer will get into the cell. Then if the more than certain amount of polymer gets into the cell, then it creates a toxicity. So that's why with the NH2 group, people have shown some amount of polymer, more than some amount of polymer you put, the toxicity is going to increase. And then if it is a COOH, then getting into then it becomes a negatively charged. Getting into the cell itself is a problem. So that's why what we selected is a neutral charged OH group polymer with the 4th generation. When you keep the 4th generation, what happens is, say, the molecular weight is around 16,000, around 15,500 molecular weight. And we do not wanted the molecular weight more than 35,000, because if this does not degrade in the body, there is a thumb rule. The kidney can process up to 35,000 molecular weight of the biocompatible polymers, or the proteins, which is less than 35,000 molecular weight. So that's why we selected around 15,000 molecular weight of a polymer, and then we try to use this as a drug delivery vehicle. So then first thing what we did was we conjugated this with a fluorescent molecule. A fluorescent molecule is fluoroisothiocyanate. FITC, it's a very well-known fluorescent molecule with a simple chemistry which we have conjugated with the OH group of the dendrimer. And then we selected the animals with our healthy rats. The healthy rats means, say, what we want to study is the posterior part of the eye. So the healthy rats is the rats which is having the good photoreceptor cells. And the diseased rats are Royal College of Surgeon rats. That is the genetically modified rat. With their birth, it has a degenerating photoreceptor cells. Okay, and after 90 days of its birth, they become completely blind. They will be suffering from retinitis pigmentosa. What that is, say, this is exactly the retina. This is these are the rod cells and cone cells, photoreceptor cells, which is held by pigment epithelial cells. So, this pigment degeneration of this pigment epithelial cells will not be able to hold this rod cells and cone cells. Because of this, these cells will also start dying. This is called retinitis pigmentosa. So the rats with RCS rats will have a genetically modified rat which have a retinitis pigmentosa from their birth. And then, what we injected was a 3 microgram equivalent of FITC, because, in the rats, the vitreous chamber is around 60 to 70 microliter. You just cannot inject how much ever we want. The maximum change which we can do is around 5 to 10% maximum change of the, otherwise, the vitreous chamber will get disturbed. So that's why we decided around 3 microliter of, extra thing we are going to inject. So the 3 microgram in 3 microliter which we injected, with a pure FITC, and another with the conjugated with dendrimer FITC, that is equivalent of pure FITC. How much it is? 3 microgram equivalent of dendrimer FITC and 3 microgram of pure FITC we injected into the vitreous chamber. So when we did this, so this is we took the cryosection of the retina. After euthanizing the animal, we took the cryosection of retina. Just look at this. This is the retina. This is exactly the cryosection of the eye. You can look at here at dendrimer FITC. After 24 hours, you can see that it's the dendrimer FITC is all around. So this star, this indicates exactly where the retina is. Okay, retinal cells. Just see this, the free FITC in 24 hours, it's almost nothing is there. It is washed out. So whatever we are getting, it's an autofluorescence of the tissue. Basically, very small amount must be here, and then it's totally washed out. And then you just look at here after 24 hours with a dendrimer FITC in a diseased rat, you can see the dendrimer FITC are coming and accumulating in the place exactly where the disease is. That is the retina. Okay. And you see that after 10 days, this dendrimer FITC is present there. Okay. So the free FITC in a diseased rat, you can see after 1 day, it's in assorted form. It's in everywhere. And in 10 days, the free FITC is nothing is there. Whatever you get, this is autofluorescent from the tissue. So what this really indicates is dendrimers appears to selectively accumulate on the place where the degeneration of retinal cells are taking place. This is exact place where we want to deliver drug. So why it is happening? So this is one of the theory which we have, which we are trying to understand. So what we understood from this is, as the degeneration starts, the retinal cell starts degenerating, the debris of the cell will trigger immune response. This activates the central nervous system immune response. The macrophages of central nervous system, usually the 3 important macrophages of central nervous systems are microglial cells, astrocytes, and Mueller cells. What happens when the disease starts? That means when the cell starts dying, the debris will create the activation of macrophages. Then the macrophages come near to the place where the disease is and starts releasing cytokines. And this release of cytokines, actually what happens is, this creates the nearing cells, it is going to affect them, and the degeneration of retinal cells will happen quickly. So then this creates a inflammation because of the release of these cytokines. Then the first thing what we will have to do is inhibits the activation of these macrophages. That means the release of cytokines. That itself will help the inflammation to reduce drastically. This will create a good thing. Though the degeneration, we cannot stop the degeneration because of the whatever the a genetic, if it is genetic, but further aggravation of the disease because of the inflammation can be stopped. So that's why most of the time what they give is a steroid to inhibit the activation of this macrophages. That's what exactly the drug is. And then, to check this, whether these cells are getting activated or the activated macrophages are present near where the disease is. That's what which we checked. So GFAP immunostaining of activated retinal cells. See, that's the GFAP. We put the GFAP there. It is binding. That means it only goes and binds where the activated microglial cells are present. Exactly, that is there. So when you look at that, this is the GFAP staining in a red, and then say in the green is the whatever the dendrimer is. So exactly that is there in the area. The same thing like methodology was done for the activated microglial cells and the astrocytes. This shows that the dendrimers are localizing in where the neuroinflammation is there, and the neuroinflammation is because of the activation of microglial cells and the astrocytes and the Mueller cells. What we checked is for the astrocytes and the microglial cells, which are activated or not, which this immunostaining which we found out, they are activated microglial cells and the activated astrocytes are present in the place. So next thing, what do you expect? So shall we put a steroid, a drug conjugating on the surface of the dendrimer, which is the size of around 6 nanometer, and then see whether it gives the same therapeutic effect? If that is the case, then it should go and stay there and release drugs slowly. The drug actually what it does is it inhibits the activation of these macrophages. Then the inflammation reduces drastically. That will stop further degeneration, the aggravation of the disease. So that's what which we did by using the chemistry. Use the spacer, and then this is a pure chemistry which we worked on, and then drug was conjugated. We conjugated using the ester bond, because ester bonds are what we want is it goes there and it has to degrade. The bond should degrade. So we made a covalent bond with the ester bond. Ester bonds are known as hydrolytically cleavable, so that though it will not be cleaved at a time, and then it cleaves slowly. So that's what which we did. We made a ester bond which are hydrolytically cleavable, and then we characterized it. The characterization we put around. See, the dendrimer, he is a water soluble. This is a water soluble because there are a lot of OH groups are there. Steroids, as we know, it's a highly hydrophobic molecule, and we have optimized it around 5.5 molecules, approximately at average of 5.5 to 6 steroids. If it is conjugated on the surface of the dendrimer, then it keeps the solubility. If it is more than that, then it precipitates. So what we want is it has to, when we inject into the vitreous chamber, it should be solubilized in the vitreous chamber in the solution, and then it should float and go to the posterior part of the eye. So that's why we conjugated around 5.5 molecules average dendrimer on the surface of the corticosteroids on the surface of the dendrimer to keep the solubility, and then characterize with the NMR, and then it is further checked with the MALDI-TOF to check the molecular weight, whether it is matching. And then we also released this in a say 7.4 pH. That is the vitreous chamber. The pH is 7.4. So if it gets in to put in the vitreous chamber, say how it releases. See, when you look at it at 7.4 pH, the dendrimer releases up to 3 months. It's a very good. And then say to compare that, we have say PEG, polyethylene glycol, which is a polymer, which is a linear polymer. We conjugated the same way and checked it it is around 30 of rather around 50 days, around 90% is released, and here it is in the 3 months, it's only 60-65% is released. So that means this is dendrimers are releases much slower than how the linear polymer is. There is one more theory based on that, because the dendrimers are a dendritic structure. Some of the enzymes are required to cleave, or in the process what happens because of its structure, the release is very slow. That is one more analogy which we have understood. Then afterwards, what we did was we took a RCS rats and for the experiment, and then we have to check how the retinal cells are active or not. That's what the main goal is. So the important way with the evaluation is electroretinography. Electroretinography, that is ERG. It shows how active the retinal cells are. The electrical signals processed by retinal cells. If the signals are more processed in the retinal cells, that means more cells are active. That's why it gives electroretinography data. The other one is you take then check the how much cells are active, the cell count by in outer nuclear layer of retina. That is where exactly the disease is. And we injected right eye was injected, that ocular dextrous, and left eye is kept as a control. Okay. The experiment was conducted between 5th week to 9th week of their birth. So, as I said, 3 months this becomes blind. So it's almost like a 1 month of their birth to, second month of their birth. So where the disease is in a prime state. So that's what all the rats were used. So with the birth, and then take them in the same time, the 5th week to 9th week. So we look at here the 5th week to 9th week. So electroretinography data. So ERG amplitude is more, means the more cells are active here. You just see that as we expect, it decreases slowly, 5th week to 9th week. So, this untreated, the another is a PBS as a control. Just look at that, it decreases slowly. Okay. So then the same 3 microgram equivalent of drug is used. You can look at that, the dendrimer, when we injected electroretinography data, it is a much better one. So in the process, what it shows is, it's say ERG is, when you look at that, so 1 month, one injection of 3 microgram equivalent of drug, it can keep at around, 1 month. Oh, this is a very good, data. And then say, when you count the cells, you can look at that, not even 3 microgram. So 3 microgram injected, and see if the 1 microgram equivalent is sufficient for 1 month, the number of cells which are active is much higher than whatever the implant which is present, which releases 0.2 microgram per day, which is present in the market. So if you compare that, around 6 times lower dosage is sufficient to keep the cells which is more active than what is present in the market. So what this really shows is sustained intracellular release from the dendrimer is possible. Okay. So now I will, this is the whatever which I explained. This is the conclusion of that. One injection of 3 microliter of injection with a 1 microgram equivalent of drug and 3 microgram equivalent of drug, which is sufficient to keep the ERG data almost stable. That means to keep the retinal cells active. And then, the same, if you use the same drug, with that, the number of cells which are active is much better than the whatever present in the market. So now, see, this is why it happens. So probably the dendrimers are taken up by the cells with a different mechanism compared to the free drug with a different mechanism. Rather, it is an endocytosis. So endocytosis is the one when it is this, the macrophages are very well known. Whenever the nanoparticles were near to that, it starts engulfing it, and it starts engulfing, and then once it engulfs, then the drug will, nanoparticle will be there inside the macrophages, and it releases drug. Once it releases drug, that inhibits the activation of the microglial cells, which activation will release the cytokines, and this will reduce the inflammation. The same thing what happens in cerebral palsy. Cerebral palsy is one of the things which is a major problem. There is almost a no cure. With the birth, somebody will get cerebral palsy, and then first thing what they will have to do is that reduce the neuroinflammation. So when at the birth, after 1 day, if you inject a particular place of the brain with this polymer, it goes and inhibits the whatever the inflammation is. That means the macrophages, it goes and gets into the macrophage and releases slowly. So this is again done with the cerebral palsy. This will be useful in that also. So how the nanoparticles, which are very useful, because steroids right now people are taking through mouth, some cases. You just imagine from the eye drop, 2 to 3% gets in. If people take in through mouth, and it's supposed to reach the place, probably 0.2, 0.3% it will reach, and the steroids has got tremendous side effects. So that's why people, for say age-related macular degeneration, they do not take any medicine, because you cannot administer steroid for such a long time. The side effects are so high. You assume, say, some cases like age-related macular degeneration or retinitis pigmentosa, which the retinal cell starts dying. Somebody at the age of 60, he gets, say, because age-related macular degeneration, let us assume age-related macular degeneration, the macula is the concentrated cell of retinal cells. The macula is degenerating, assume. And because of, once the degeneration starts, then inflammation occur. Because of that inflammation, the degeneration gets aggravated. If we stop inflammation, then the degeneration will be very slow. Say somebody is going to become blind at, say, 65. If he becomes blind at 75 or 80, then you are giving a 10 years of a good life for a person. Now they are not doing it because you cannot give a steroid through mouth or through the eye drop. It hardly reaches there. So if you inject, say, every 6 months once, something like that, and then exactly it goes there and stays there and releases, this will be a very useful drug delivery vehicle. So nanomedicine will be very useful here. Now I shift to something, cancer. How can nanomedicine, nanoparticles, and through nano, how the cancer will be, treating cancer will become easy. So the cancer is, as you know, it is a uncontrolled growth of the cells. Rather, because cancer is something which happens, some control which supposed to follow by our own cells, which is not following it. That is the cancer. So that is that we do not have control over our own body. That is exactly what is the cancer is. Okay. So uncontrolled growth of the cells because of lot of things. And then if you put a nanoparticles, the drug cancer cells, the cancer drugs are what? Basically the drugs are which are going and kill the cells which are multiplying. Okay. So that there is nothing called the drug will go exactly the cancer cells and then it kills the cell. Whichever cells which are multiplying, it goes and binds there and stops multiplication. So that's why in the process, the side effects of the cancer drugs is tremendous. How to reduce these side effects? This is a nanomedicine will be useful. That is exactly what we are looking into. So cancer, as I said, it makes cells which are uncontrolled growth. Or I would put it in a very simple way. What the cancer is it is just like in our society. What who does the people who are antisocial elements who do not follow the rules. Okay, the cancer cells are like that inside our body. So they're supposed to follow certain rules and they are rebellious. They are not following those rules. So that what is that rules? They have to get into apoptosis, and then the programmed way of cell death should happen. But they do not die, and then it is just like they starts growing, and cancer cells are jack of all and master of none. So once they start growing, obviously they require nutrition. When they require nutrition, what they have to make their nutrition through, they have to create their own blood vessel from the regular blood. It is just like, it is just cancer is, I could connect with like a slum, and somebody in a slum, they want a current. What do they do? Whatever the current line is there, they connect it to them. But it may not be very proper way, but they connected it. In the process, it may be very dangerous also, same way. And the water pipe, and then somebody connects the water pipe, which is the regular pipe. Same thing happens inside our body. So since they are jack of all, they have to survive. Then they make their own blood vessels from from the actual blood vessel. They create their own blood vessel to get the nutrition. While making that, the blood vessels are leaky. So they cannot make a good blood vessel because it's not there. They are jack of all. It's a survival instinct, but master of none. And then in the process, what happens? If it is a nanoparticle passes through where the tumor is, because this is the tumor, when the tube passes through the blood vessel, it is a leaky. The nanoparticles will get into the tumor very easily, and they do not. And then afterwards, there is something called in our body, blood is the nutrition it follows, and then another one is a lymphatic drainage system, and it's a poorly connected lymphatic drainage system. So once the nanoparticle gets in there, there is no lymphatic drainage system exactly present properly. In the process, what happens? The nanoparticle stays in the tumor for a long time. And one more thing about this cancer cells are, as I said, they are jack of all. They overexpress certain receptors, like certain integrins are overexpressed, like vascular endothelial growth factor, because they have to vasculation, they have to make vascularization between the other thing. They overexpress certain vascular endothelial growth factor receptors, which are overexpresses that, and then epidermal growth factor receptors, they overexpress that. In the process, because of this overexpression, you can create a ligand on the surface of the nanoparticle. So suppose a receptor, for that receptor, you create a ligand, the nanoparticle encapsulated with a drug, it goes and binds to that receptor and taken up with the mechanisms called receptor-mediated endocytosis. So this is actually happens in some of the cells. So then when it takes up in that way, then it releases drug. Then this becomes you are targeting a cancer cells. Not exactly, the without the targeting moiety is a passive targeting, and with targeting moiety it is a targeting to the cancer cells. That means since it is not only cancer cells expresses, the cancer cells overexpresses. So because it overexpresses, the chances of getting nanoparticle with a ligand on that is much more than the getting into other cells. So this is called enhanced permeation and retention. It is called EPR effect. Enhanced permeation will be enhanced, and because there is no lymphatic drainage system, retention takes place. Because of this, then if we do this with the nanoparticle, and then we created a on the surface as RGD peptide sequence, which is a ligand for alpha V beta 3 integrins, which is overexpressed by some of the breast cancer cell lines. Okay, so if you put this and then we checked into the thing, you just look at it is here, how much a cell it will take up. You can see this, the nanoparticle taken up with the CRGD peptide is much higher than, say, you put a CRGD as a excess outside, and then you just look at that here, it is much less. Instead of CRGD, we modified whether, because the ligand is a perfect, it should be CREAD. That is, C is a cyclic, RAD is the peptide amino acid, 3 amino acids which we have put. If you put the CRAD, you just look at here, the amount of taken up with the receptor-mediated endocytosis is much more than the CRAD. So this, again, we can see the same results in the tumor cells. So the tumor, if we look at how much it is, the same results which we have got in the tumor. So the with the ligand on the surface, more nanoparticle gets into the tumor cells. The same thing which we did with the epidermal growth factor receptors. It is overexpressed by epithelial cells, cancer epithelial cells. And then when we, this is a particular ligand which goes and binds to that receptor. So using a chemistry which we created. And look at here, the same results which we get, that this PANC cells and then A549 and U87, all of them are which overexpress EGFR, and then you just look at that, the amount of intake into the cell with the ligand on the surface is much higher than without ligand on the surface. So then how the killing is, whether with the killing is in the same way, just look at this, it is at the 10 nanomolar equivalent. Just look at that, the cell viability, it is almost 30% much higher than the cell death compared to the nanoparticle, and then compared to the free drug, it is much higher. Okay, so we did this with the A549 tumor. We grown into the mice, nude mice. Look at here, it is with the nanoparticle and then EGFR, the tumor growth is, it slowly grows compared to the control and the drug. Okay, so the drug will be more effective if it is into the nanoparticle and if it is put on the a ligand on the surface of the nanoparticle. That's exactly which we have found out. So the cancer, the main problem with the cancer is, most of you know about it, when metastasis occur in the cancer. What is this metastasis? Metastasis is, when the tumor cells, I will put it in a very simple term, when tumors grows beyond that, among the tumor cells, some of the cells are very smart and they understand why I have to struggle with these cells. Let me move out of from among these cells and go out and somewhere, let me re-establish somewhere else. So that cells are a very, smart cells, and they have stem cell like property. They're called cancer stem cells. They move out and re-establish, move out and establish in some other place. That is called metastasis. When metastasis occur, then that is the problem. Curing the cancer is very difficult. Then, which is these smart cells? We will have to kill these smart cells. Okay. So the smart cells are called cancer stem cells, which has a stem cell-like property. Say, suppose if you kill all the cells, it reduces. If it is only one cell which survives, then the tumor will grow again. This is exactly what happens. So now, like how to target the cancer stem cells are by making the nanoparticle, whether it will be useful. So what we have understood is with the epidermal growth factor receptor ligand on the surface, when we did that and checked the amount of cancer stem cells, just look at here the amount of cells which has got a stem cells. You put some amount of drug. What exactly it means is here the amount of cells, when you put the drug, the cells which are cancer stem cells like property, the percentage is going to increase because they have the ability to survive. Okay, so we use a 5 nanomolar amount of paclitaxel. You just look at that in the nanoparticle, it's around at 25%. Say control cells, usually it's around 4% of the smart cells. If you, after the treatment, when you look at this, just see the cancer stem cells population should increase as it is. Say that has increased because those cells are surviving. But if you look at here, when you put a targeted moiety on the surface, then the amount of cancer stem cells are also reduced drastically, significantly. This shows through the receptor-mediated endocytosis, when a cell takes up, there's a chance that it evades the cancer stem cells for the survival. Okay, so what is this cancer stem cells? They have something called a PGP like proteins, which has affects the drug outside when the drug gets in. So probably when it gets through the receptor mediated endocytosis, it avoids that efflexing pump, and then in the process it will be able to kill the cancer stem cells. So further work is required in this area. And now the many people have worked on this. The antibodies are produced, especially for some of the blood cancers. The cure is there. Actually, the antibodies are produced, which goes and binds exactly the cancer cells because of the certain receptors. It goes and binds exactly to the cancer cells, and then neutralizes the cell, and it is there in the market right now. That's what exactly with the nanoparticle compared to the free drug, nanomedicine will be very useful to cure cancer. So now the last part, which I go into tuberculosis. The tuberculosis is a very a deadly disease. It's a 6,000 years old disease. And then, more than 100 years back, Robert Koch, which he has identified it. And first vaccine, BCG vaccine, came in 1920. Okay. But still there, it is only for the juvenile people up to less than 5 years. It helps. Still, there is no vaccine for tuberculosis. And then, Southeast Asia is a, breeding ground of this tuberculosis. 30% of the world's tuberculosis patients are in India. And then, as all of you know, our Prime Minister has said by 2025 we should eradicate the tuberculosis. I don't know on what basis he said this, but WHO has kept by 2040 or 2050 we should eradicate tuberculosis like smallpox and other diseases from this world. That's what the WHO's idea. Is it possible? That's what the present treatment, whatever it is it possible to eradicate? That is the questions which we are going to ask. So the problem is that tuberculosis is, this is the data exactly how many people are in India and what are the number of people in the globe. So in India, around 32% of TB deaths happens in India, and 27% of all TB cases the world is in India, and 24% of multi-drug resistance TB is present in India. So why this is very important? We will have to understand the pathogenesis of this disease. Just look at this. This is it's a through aerosol. The tuberculosis is a very small organism. It spreads through aerosol. Say somebody gets into a hospital and then you inhale with the patient's mouth, and then that's it. You are infected. Infection is not a disease. That is one of the things what you will have to understand in tuberculosis. Why it is so? Just look at here. When you inhale something, it gets into the cell, that is the lung alveoli cells. It gets in. Once the problem with that is the macrophages, alveolar macrophages, identifies the tuberculosis quickly, and it engulfs, or it in a simple word, it swallows. That's what the macrophages do when it sees any pathogen. When it swallows, it will not be able to kill that, because how it happens is macrophages, it forms a endosomes. From endosomes it gets into lysosomes, and lysosomes it lyses each of those, and then it gets into cytoplasm, and then the proteins will be processed. Or macrophage take the particular protein and goes to lymph node, and then if the macro, and then gets into to give it to B cells and T cells to produce identifies to produce the antibodies for that particular antigen. This is how exactly in the case it happens. In this case, what happens when it gets into endosomes, it will not able to kill, and lysosomes it will not able to kill, because mycobacteria tuberculosis, the cell wall is made up of mycolic acid. It is resistant to the whatever the killing which happens with the other organisms. Then what happens? This macrophages gets starts. It is just like it starts giving a signal to the other cells, other immune cells. Then those immune cells, what they do is it comes and makes a small ball-like structure that is called granuloma. So what it has is the macrophage is inside, and it forms a ball like the other cells is around, and then it forms like this structure, and then this is 50% of Indians. That's what Wikipedia says, but the physician says around 70 to 80% of Indians will have this. They're infected, but infection doesn't mean that it's a disease. What happens is when it forms like this small ball, that is a latent tuberculosis. It survives inside, but it will not multiply. It survives there. When the immune system is in compromised condition of our body, then what happens? This latent tuberculosis, it slowly starts multiplies and gets out of the ball-like structure of granuloma and spread to the other part, or it multiplies in the other part of the lungs. Now you want to kill, or you want to put a drug for this, the prophylactic effect. That's what people have tried. You want to give when this is there, say when it is an infection, you give the drug to here. The amount of drug reaches here is so less. The side effects of the drug is so high because you have to give antibiotics, and then the prophylactic effect will not work. And once it forms this, then you start giving the drug. And then tuberculosis, Mycobacterium tuberculosis is a very a smart microorganisms. So since it is very smart microorganism, we have to give 4 types of drug to confuse it. Okay, so how the drug treatment is, the 2 months you give a 4 drugs, which how the drugs work. There are, say, rifampicin, isoniazid, 2 important, very important drugs. That is isoniazid acts on the cell wall, and then this cell wall degradation starts. Because of that, starts dying. Rifampicin, it goes and binds to RNA polymerase of the then it will not able to produce RNA synthesis will not happen. That's why it is going to die. And say same thing with the pyrozinamide and ethanbutyl. So initially you give 2 months of these 4 drugs. Finally, you give 4 months of rifampicin and isoniazid. So 6 months you have to take drug. It's a very long process. So in the process, what happens? Most of the people are uneducated people. After taking 2 months of the drug, they may stop taking it because they may start feeling better and stop taking the drug, and then survived one will get a resistance of that. Because of that, multi-drug resistance TB is very common in India. And now extremely drug resistance TB is also coming up. That is also common in India. So now how to solve this problem? Okay. So the granuloma, when formation takes place, there are 4 stages. One of the important granuloma stage is necrotic granuloma. In this, what happens is, though the granuloma is intact, it starts slowly, some activation of the latent TB will start taking place. If you identify during this time and put the drug here, and drug should get inside the macrophage and then stay there and release drug for a long time and then kill. Instead of, the burst open and then it goes to some other place. If we can target here and cure the disease, then, it will be a great help. So the initial work, what we did, that's which we are going to explain. So this is exactly what it is. Say the macrophages takes it, and then suppose we put the drug with loaded nanoparticle with here, and it's the nanoparticle stays inside the macrophage and releases. And when it releases, the chances of killing if it releases for a long time and killing is very high. And for this, we use the 2 types of drug, rifampicin and isoniazid. Those are the drugs which are used. It kills the 99% of the TB, and then very potent drug. And the problem with these 2 drugs is you test to when you're taking, as I said, the last 4 months you're taking these 2 drugs simultaneously. There is a chance that there is a reaction between these 2 drugs, and some of the drugs will not be used at all. Okay. And the other part is what we have identified is isoniazid has got a NH2 group here, and then it's because of this, it's a highly water soluble drug, and the drug penetration into the cell is because tuberculosis survives inside the cell. Drug penetration itself is a problem. That's why the more drug is required. So what we have thought was, can we make it a little more hydrophobic and make it more stable and gets into the cell easily? So we modified this with benzaldehyde. Benzaldehyde is a common food supplement which is used, and then which we made this with the benzaldehyde, a small modification, and used and checked whether it has the same potent as isoniazid. And rifampicin is, as I said, it is tested as a rifampicin. As I exactly said, each of these drug independently they can kill. Okay, and also check the how the rifampicin stability is. You know, rifampicin highly unstable at acidic pH. You can look at that in acidic pH. One is at the room temperature, other one is at the body temperature at 37 degrees centigrade. At body temperature, you just look at that, it degrades. Same thing at 4.5 pH, it degrades. And then at 2.5 pH, it degrades so quickly. Okay. So that's why, you know, after the food, you will have to take, because, you know, if you take the food when our stomach is 2.5 pH, it's before the food, it's almost hardly you find the drug reaches the actual place. So this is what the stability of the drug which we have identified at a different pH condition. And then we released in the nanoparticle whether if you put it in a nanoparticle, it is going to increase the stability of the drug. That's exactly what we found out. You look at that, the drug will be released for 30 days, except the 2.5 pH. 2.5 pH, the nanoparticle cannot hold exactly because what we use is a polylactic lactide co-glycolide polymer. At 2.5 pH, it is made up of a ester bond. It breaks open. So that's why you look at here that, you know, it breaks open, and then at 2.5 pH almost, you know, it's not a release. But in other cases, you can look at that up to 30 days, the drug is available. So we measured, the measuring of the drug is the intact drug, how much it is. So that means that by encapsulating into the nanoparticle, the stability of the drug is going to increase drastically. Then we checked with the same way how the saliva or any patient's you know, infected patient is checked. The MGIT BACTEC 960 instrument, this is the WHO approved instrument. All the, you know, this is the gold standard in the case right now. What we use is, we use a 37RV strain. That is one of the tuberculosis strain, which is incubated and kept in exactly in this instrument. And then we added our whatever formulation in nanoparticle and comparing that with the control. Okay. So you just look at here, 1 microgram equivalent of a rifampicin, actually the growth unit is 0, means it inhibits the complete growth of tuberculosis. That means 1 microgram per ml equivalent of drug reaches the particular place that will kill all the tuberculosis. Okay, that is called a MIC, minimum inhibitory concentration, for its growth. Then afterwards we checked whether it only inhibits or it kills. It actually kills. And then we just look at that. When we reduce that to in a nanoparticle around 0.7 microgram equivalent of that. Okay, so 0.7 microgram equivalent of nanoparticle in the form of new formulation, it inhibits. So almost a 30% of drug is reduced by just encapsulating into the nanoparticle. Say 0.7 microgram equivalent of say rifampicin, you can guess that it will not. So the growth unit 400 in the sense that exactly at the growth of tuberculosis. So 30% of the drug can be reduced just by encapsulating into the nanoparticle. So once we modify with the isoniazid, now when we look at the isoniazid, we modified the isoniazid with benzaldehyde. We call this as IH2. So we are the first people who showed this new pro-drug. Actually, you can call it as a pro-drug. Just look at this. When we make that, the IH2 stability is going to increase drastically. At say 7.4 pH, just look at this isoniazid degradation occur, and this is the IH2 degradation. Though the stability of IH2 increases drastically, and the IH2 release is also, you can look at it at the different pHs at 7.4 and 4.5 pH. As I said, this 2.5 pH is very difficult to make a controlled release. And then see, you just look at it, the 7.4 and the 4.5 pH, you can release slowly the IH2. And one of the other thing which we have identified is encapsulating into the nanoparticle increases drastically, around 15 to 20 times increase. If it is isoniazid is a highly water-soluble drug, if we only want to encapsulate into the nanoparticle like a hydrophobic polymer, most of the polymers are hydrophobic, and then we want to encapsulate, then only 10 microgram is possible. If you modify it, it's nearly around 175 microgram per milligram can get encapsulated. So it's around this 15 to 20 times much more than the actual encapsulation. And in the process, the amount of drug availability is also going to increase. Then we checked how exactly this is going to. This one, oh sorry, I think I have missed one. Yeah. Okay, then when we check that with the IH2, we get the same effect as isoniazid. So when you modify the drug, the equivalent of a minimum inhibitory concentration of isoniazid is actually 0.1 microgram per ml. Now, when you modify that, molar equivalent, it will become 0.16 microgram. If you put a 0.16 microgram of that into this, it inhibits the total multiplication of tuberculosis pathogen. So it acts exactly like a drug with the modification, though we were not able to decrease the concentration. So it acts like a drug. If you reduce the concentration, the minimum inhibitory concentration is not going to decrease for the whatever IH2. Now what we did was, the macrophages are, as we said, these macrophages, when we get into the cell and then it should release the drug. We did a simple experiment. What we identified, all the macrophages, most of the macrophages overexpress folate receptor. So for that, the ligand is folic acid. So we put the folic acid on the surface of the nanoparticle and checked on one of the macrophages, a RA 264.7, that is the cell line, a macrophage cell line which is available. You look at here, the folic acid, folic acid conjugated one, compared to the PLGA, only pure drug, the big difference. Okay. When you put a PEG on the surface, it increases a little bit. There is one more theory, because PEG avoids the macrophages to opsonize. So what we put is on the PEG, we put the folic acid on the surface. You just look at that, this is the nanoparticle, and then increase. Whether the folate receptor is acting, what we did was we checked by adding some amount of folic acid, in pure folic acid, to block some of the receptor. You can look at here, to block some of the receptor exactly which is blocked. And this is with some amount of folic acid, free folic acid. When we put, the uptake of the nanoparticle in the cell decreases drastically. Okay, so now the next thing is we can also make a folic acid on the surface. We can target the macrophages so that in a tuberculosis with the folic acid on the surface, it exactly goes and binds to the macrophage and then gets into the cell, and then the killing will become very easy. The problem is because the tuberculosis is a highly infectious disease, we were not able to make an animal model and progress in this project. It requires a biosafety level, a higher level. So similar type, what we did, say chlamydial infection is a deadly infection which survives. The chlamydia survives in intracellular. Just look at that. If a nanoparticle will, because it survives, say this is a chlamydial which survives. If you put a nanoparticle, you just go see that nanoparticle gets into the chlamydia. The chances of killing is extremely high. So for infectious diseases, many of the infectious diseases like this, the nanoparticle will become it also, because nanoparticle will also gives the stability for the drug, because at a time the drug will not be exposed, and then slowly when it releases, the amount of therapeutic amount is going to decrease drastically. This will help in killing the infection, and also reducing the drug resistant pathogens. So I stop here. The first work which I did it in Wayne State University. Right now Dr. R.M. Kannan is in Johns Hopkins University as a professor. The second work with the cancer, which I did it in University of Minnesota. Right now, Dr. Jayant Panyam is Dean in Temple University. And tuberculosis work, there's a first graduate student, Dr. Shushruta Akimani. Right now, she is a research associate in the same department. So this is the tuberculosis is it is my idea. And then some of the work like a folic acid on the surface. This is done by Mr. Sri Prasad Acharya. He's also doing PhD with me. He's an assistant professor in chemical engineering department. The targeting, though I have not shown, we are trying to target the cancer stem cells. Not much of a good data produced. One of my PhD student is working on that, and one of Joyce Pinto is my MTech student who worked on linear PEG with conjugating with the drug and see the effect. And I also collaborate with Dr. Santosh Gaonkar of Department of Chemistry for chemistry part of it. Say if I get an idea whether this chemistry works or not, that's what like a benzaldehyde conjugating with the isoniazid, he came up with that. So the same with, we have also conjugated with so many other things. And Dr. Bharat has done a bioinformatics works of that, and recently we published that which one is more efficient and which one is less efficient. We came up with certain things and recently published. And Dr. Bharat did the bioinformatics part, why that is a more efficient, that part. And Dr. Vishnu Prasad is a Department of Microbiology in our Kasturba Medical College. He because similar to patients how they treat the patient, similar way we wanted to analyze. So he is the one who is authority in that. And Dr. Indira Bairi, she is also microbiologist in Manipal, Malika Manipal Medical College in Manipal. So I collaborate with all of these people, and I also collaborate with Dr. Guhanjaya Raman, though none of the I have not included any of the slides here, whatever our collaboration is. And also collaborate with the ophthalmology department of the KMC to further whatever I found out with the dendrimer, can we do it with the different drug and more appropriate for Indian condition. That's what which we are looking at. Thank you, and I am very happy to take your questions. Hope you enjoyed my talk and something whatever which you have learned from here. Did I take a yeah. Hello.
Hello.
Hello. Hope I have not put all of you to sleep.
Yeah. Yeah. No, sir. It was really.
It's almost 1 hour 10 minutes. Yeah, I'm sorry about 5 to 10 minutes
No.
of extra. Yeah.
No, sir. Not a problem. Yeah. Thank you very much for your very insightful talk, sir. And this is the time to take a questions. And I have good number of questions from audience, and I would like to pick a best of the best questions. And prior to that, I would like to take a questions from panel members if there are any. And if there are any questions from the panel members, I request you to ask the question.
Bharatji, I think
Yes.
it was very refreshing to hear to a chemist among this, because all this time we have been listening to only biologists. And it was
Okay.
a fantastic presentation and a field of work where we are also interested and we are doing some work. So I'm sure Dr. Lata will be excited to ask you questions because she works with chemistry of medicinal chemistry of natural compounds, and Dr. Shiban, who is very keen on tuberculosis. Over to you, Dr. Shiban.
Yeah.
It was Doctor. It was fascinating to listen to you. It's all fundamental work, and hopefully you will find applications to tuberculosis fast, quick.
That's what which we are trying. We patented it, and then we wanted to take it further, but it is very difficult, especially animal model. We have to take it to animal model, but the animal model, the main problem is biosafety level. We do not have in Manipal, and I think only 2 places in India they have, and that's where it has stopped. And we want to take it at least to the macrophage level, infect with that, and modify what all cytokines it produces, why the gene changes takes place. I mean, a lot of things can be done.
Yeah. Are you aware of any work being done in the TB institutes of India regarding this or anything new that's coming up?
Right now, I do not know. Actually, this is whatever it is, a 2 years old, 2018. Afterwards, we wanted to continue with the macrophage infection and all, but nobody was ready to take it because though we can have a non-virulent strain, but when it becomes virulent, we really do not know. And I am a chemical engineer, so I wanted a microbiologist to take care of this. And for that, the biologist, we said we will grow the macrophage and give it to you. You do the infection, and then we will give the new formulation. You check it. But the problem is that even they are also not interested because there is the animal cell line, and then they contamination. There should be a separate place for it, and then for that extra money is required. A lot of these things. So in the process, we were not able to take it to the next level. I really want to take this to the next level. And then right now, as you had asked, one group in PGA Chandigarh has done the work, a lot of work. I think the professor has retired. He was in biochemistry. I think Professor Kullur of PGI Chandigarh. They have done a good amount of work in that, and then afterwards, nobody is taking up of the next level.
Yeah.
And recently, I saw one of the papers from the PGI Chandigarh only, because tuberculosis, eye tuberculosis is I have never heard of it. Eye tuberculosis is a deadly, it seems, because they really do not know how it has spread to the eye, and then exactly where it is a retinal pigment epithelial cells. What I said that the first model, that's where it gets intracellularly, it survives there, it seems. And then like a tuberculosis means orally people take, and the amount of drug reaching there is very small. And if somebody makes an animal model, I will be very happy to inject into vitreous chamber, and then we can. If people come to know this is a eye tuberculosis, we can easily cure with whatever technology or whatever we have developed. That is one of the papers which I see, not with the nanoparticle. If it gets into that, what all changes it happens, like a cytokine, if it is a tuberculosis is infected. That work is done in the PGH Chandigarh. I saw that recently. Other than that, I think people are trying to make a vaccine, and I feel it's a very difficult, because it is not like our macrophages will not identify. They identify it, but they will not be able to kill that. So once it gets into that, whatever in that ball, can we deliver drug to the ball, say by inhaler? If you create a inhaler with a nanoparticle, we inhale the nanoparticle and it goes to exactly to that place, and to the granuloma, and then gets in there and releases drug slowly, and we can kill that without spreading to the other part. That will be the great thing to happen. My understanding, whatever about this.
Yeah, it's kind of frustrating. Those of us who work with tuberculosis, not a single new drug has come in decades. The drugs
Yes,
you mentioned
yes,
are,
correct. It's a 1960 rifampicin and isoniazid. Both of them are 1960s.
pyrazinamide also is an old drug. You can see
Yes, yes, correct.
outside every hospital, see dead grass. When you see that, means the people have sprayed pyrazinamide there. Tuberculosis people took it, free
Yes,
pyrazinamide, and just threw it out. It's kind
correct.
of sad that we haven't got a single drug in decades for this. Anyway,
Yes.
and that was my only question. I thought you'll give me some exciting news that a new drug is coming, but I think we've got to wait. And
I'm not in a medicinal chemistry, so
yeah,
I'm sorry about it. I do not know much about it, because my is a drug delivery. I work on drug delivery, so that's why how to stabilize the drug with the existing drug.
yeah,
Actually, that's exactly what with the azithromycin, say rifampicin and isoniazid.
yeah,
If you take it, they take it together. In fact, they
yes,
react. Say rifampicin has got CHO molecule, and isoniazid has got NH2. At certain condition, these two at acidic condition, these two reacts and form one more compound, which we showed and not yet published actually. And then in the process, what happens is the amount of drug required, finally what you want is a therapeutic amount. And say isoniazid is a neurotoxic. Actually, it is not so good. If it goes to the brain, it is a highly toxic, some of the things. And then it's a highly water soluble. Luckily, it will not cross through blood brain barrier very easily, because it's highly water soluble. You cannot take isoniazid for a very long time. And in the process, how to stop the drug, say what I understand with the basic thing is, tuberculosis, the problem is the cell wall of the Mycobacterium tuberculosis, because once it gets into the endosomes, with the enzymes and with that pH, it survives. That's why the macrophage gets troubled, and then suddenly it calls the other cells, and then it gets into the lysosomes, and most of the bacteria will be lysed, but here it survives. So it should be some drug which should come. Say 3 of the drug, it is related to cell wall only, and only rifampicin is like any antibiotic which is used to go and binds to RNA, to stop the RNA polymerase, to make the RNA. So otherwise, 3 drugs are good. Other than the more drug, how to make it more effective? That is a very important thing. And then see, now the XRD, you must be knowing, the extremely drug resistant tuberculosis is also prevalent in India. And there is no way to treat. I think in 4 or 5 years back, a 12th standard CBSE topper died of a tuberculosis in Bombay. Then he was died because of extremely drug resistance he was infected with. It's a what I understood is most of the paper which comes from Europe or this, we do not do fundamental research. Basically, we are more interested in producing paper. So that's what which I see. Most of the people are more interested in, say, producing more paper rather than taking it to the next level. So probably incentives are there to producing more. They have to change how the incentives say. I do not know. I am in an academic institute for teaching and also to train the people. The research institute, exactly research institutes which are there, like a CSIR research institutes. Their main goal is not to produce paper. So people should not get the promotion because you're producing more paper. Solving the problem or producing new things which should be based on that. I see like everywhere people produce more paper rather than taking it to next level.
Right. Thank you very much. And I'm done. Bharatji, Dr. Damle, I'm done. So anybody else wants to button, I'll mute myself now. Thank you.
Hello, sir. It was a wonderful session, and you have elaborated different mechanisms of your experimentations. I have a doubt regarding one studies indicated in Ayurveda Bhasma, Kasisa Bhasma, there is a presence of spions, super paramagnetic iron oxide nanoparticles.
Okay, yeah,
So,
very good.
in the raw material, we are using the iron only, and after processing, in the final product, we identified spions. So, what is the relevance of spions? I am keen to know, keen to hear you about the presence of these spions. What are its merits and demerits?
A lot of paper, yes, I got your question. A lot of paper in cancer has come in this. Actually, what happens is one or two paper, whatever read, I have not worked in it. Say this, if you are nanoparticle outside, you are through magnetic things. If you are in the blood, if you are pushing towards the tumor, it gets into the tumor. And the other part of it is it holds the heat for a long time. In the process is if it is at because our body temperature is 37 degrees centigrade, and then if it stays there and it gets heated up and it will reach to 41 or 42 degrees centigrade up to. So in that heat, actually what happens, the cells will not survive. It's going to kill because of that.
Okay.
So that's the one of the things what I have read with the cancer thing. It must be true. It must be true with the infection also. In the infection, it will be true. You can look into the things. Our body response is like that. When we get an infection, first thing the body response is the fever to reduce the infection and increase the blood flow.
Yeah.
So then in the process, what happens if you put this, the temperature of this nanoparticle, if it gets into the, it binds to the, infectious something, and then, the temperature is high because it holds more heat compared to the particle.
So it produces hypothermia.
Yes.
Okay, fine.
So it makes sense what I said.
Okay, fine, very fine.
Yeah.
Thank you for your clarification.
Can I come in, sir?
Yes, madam. Yeah.
Yes, Bharatji. That was really a very fascinating session. Thank you very much for that. My interest being in the, drug development and formulation development is whether we can improve the epidermal absorption of the topicals, one thing. And while doing that, is it necessary, because in your session I saw you had to make an ester of steroid to attach to the dendrimer. So is it necessary for us to change the formulation accordingly, or you need to customize according to our formulation, the dendrimer?
Yeah, it's a good question, madam. So one of the thing what it is because I do not study how the drug is go and bind and which part of the drug is more effective, how exactly it inhibits the activation of macrophages. So we know that steroids which are go to the macrophages, it inhibits, that means the cytokine release will be stopped. That's what exactly happens. We take the steroids and then the swelling is going to reduce. Okay, swelling is because of the release of cytokines by these macrophages. Now what we want to do is how it happens is that is not my things. Probably Dr. Bharat, if it is some people with the bioinformatics, it goes and binds exactly in that particular place and does this. What we want is we make a pro drug. Say if we conjugate with the dendrimer, and finally it should release to become a drug. That is our main goal. So if that is the case, suppose I make a amide bond. So amide bonds are very stable bond, and it is not cleavable in the actually inside the cell. Probably it will with the peptidase enzymes. Because it's a peptide bond in our stomach, it will be cleavable. So somewhere in the eye, maybe that enzymes may not be there to cleave. So that's why if you put in a water itself, if it is that enzymes are not there at that place, in the water itself, it breaks open. Then, it becomes a pro-drug will become drug. That's why which we made which is a cleavable or not. So whatever I showed the release, which is shown in the HPLC exactly how it releases the drug. That's why say some of the easily breakable bonds which we will have to make.
Okay, so in case, say I understand, okay, now that was regarding steroid. Now in case our product, we know the molecule, say it is some kind of small molecule. Okay. What I want to know is, you have developed a dendrimer, so is it going to be useful to be attached to any of these, or do you have to every time make a new dendrimer? Like, can I just
not necessary? No. You can directly buy the dendrimer. The dendrimers are available, polyamidoamine dendrimers. In India, it is not available actually. So the making dendrimer is not so easy. I did not make actually, one of our lab it has made, and then afterwards characterization and other things which I did, conjugating with the drug, making the pro drug and formulation, all those things which I did. Actually, that making the dendrimer is a skillful thing. Polyamidoamine dendrimer is a very well known this. Okay, and then see when anything which is on the surface, you have to make if you are making a conjugation or making a covalent bond, there are three bonds basically in organic chemistry, which are polar, which can make a covalent bond, which is a OH group, which is NH2 group, or it's COOH group. So on the surface of that, these three groups are present. That is available. Polyamidoamine dendrimer with a COH group outside, which is available in the market. It's not in India, it is not available, but in US it is available. Okay. And then OH group on the surface, it is available, and NH2 group on the surface, which is available. So since OH is a neutral, the OH will not become O minus and H plus, because it's a it will COOH. But there is a chance
it more polar.
Yes, it makes it more polar. More correct. Polar. More polar. Yes.
So we can actually choose either OH or NH2 like that. What you mean, right?
Yes, either OH, NH2, or COOH also.
Correct. Acid groups.
Yeah.
Okay. That was one thing, and also I happened to read one article about polysaccharide nanoparticles, where they have used the natural biomolecule of epigalactocatechin gallate, which is loaded with the polysaccharide nanoparticles. So I was wondering, so most of our, these are very commonly found in Ayurvedic plants actually, catechins, and so whether we can use the same to improve the drug delivery of Ayurvedic medicines. This is just a thought that.
Yes, definitely. See, the thing is, your area is good, madam, because what I understood is nobody has evaluated how the Ayurvedic formulations work. Probably if we evaluate and make it I mean, that's why the Ayush is giving a lot of importance for that. What you exactly said, it is correct. But only thing is, why I have used a hydrophobic is we want to encapsulate more amount of drug. We know the drug, that's why. And then with the polymer, which I used the hydrophobic polymer because it's also FDA approved, which I used. As you said, lot of other polymers which can be used, which are water soluble also, and some of them water insoluble. If it is water soluble, then you have to make a hydrogel because you have to make it a water insoluble. Finally, you want to make it a suspension of a particles,
Yes, sir.
which releases slowly.
Right.
So then you have to make then chemistry comes into picture.
Yes,
Can
you have
we
to make a
cross-linking?
gel-like, yes, call it
Yes. Can we
cross-linking
make
of
a
polymers.
cross-linking? Yes. And then it allows the water inside, then it swells up, and then the drug will move out from there and slowly, and then it also gives a little bit of a stability, all those things. Yeah.
Yes, sir.
Lot
Thank you
of
very much, sir. Thank you.
Yeah,
Thanks.
thank you.
That's
Thank you,
it from
madam.
me. Thank you, sir.
Good questions. Yeah.
I have a question.
Yeah.
Are there antimicrobial peptides, AMPs, or these nanoparticles, which without binding to the active molecule increase the skin penetration?
Yeah. I do not know, sir. Yes, I have seen. Yes, sir. But
Okay.
yes, but it's not my research area. Correct. People are working in it how to improve the skin penetration. But you have to think of one of the things, approval from FDA. That is, I made this dendrimer, or rather we in our group, and then we showed all the things. And then, approval of FDA is a very important thing. Why? When you put it into the eye, then finally it releases drug. What is the pharmacokinetics and pharmacodynamics of this polymer? Finally, how it metabolizes in the body, that is a very important thing. Okay, how it gets out.
Yeah,
And then
I understand that. I'm just thinking about loudly about topicals, not on the orals. Let's say arginine-rich peptides, and using them, but without they binding to the effective moiety for the simple reason that there could be multiple pharmacologically effective molecules in one compound. So, leading only one, driving one into keratinocyte may not be wise. So, are there something? Is there a concept of increasing the skin penetration?
Yes, there are many.
Yeah.
Yes. I think it's certain frequency. If you put on the screen, it can be increased. People are working. I think one of the important person is, I think Indian in US. He is now in Harvard. Dr. Samir Mitragotri. Because you put on that, they are trying to deliver insulin through that. Because if it increase, penetration is going to increase. Again, what I mean to say is, if penetration is going to increase, skin is the one of the outer layer, and then very important outer layer of the body. If the penetration, how much time the penetration is going to increase, that is the important part. If the penetration increases for a long time, then means with that some other pathogen will get in.
Yeah, I mean, I understand that, and I think
that is the thing. Lot of work is going on in that, actually.
I think also Dr. Haresh Parekh, who is in whom I met in Australia, is also working in your with your organization. They are look. I mean, they have demonstrated that through nose you can get into direct through blood-brain barrier into the brain.
Yes, correct. Through nose, it directly gets into the blood-brain barrier. Yes,
is one of that. And but I mean, in conversation with him, he was telling that it is possible for us to make it get into keratinocyte, but not get into the bloodstream. Probably we'll get him as a speaker one of these days. I was just thinking when I heard your speech, we should get him as well.
Yes,
thank you very much.
Useful
yeah, thank you very much. I will, let others talk now.
Yeah. Thank you, sir.
Yeah, thanks for the questions from panel, and I would like to take questions from audience. So I have a question from audience. The first question is, if there are any possibilities of triggering an allergic response in retina to the dimer, in the sense, is there any allergic reaction for the dendrimer in retina?
No, it is not there. That is checked actually. So it is not. It's a highly biocompatible. There is no trigger of response, immune response.
Okay. So
anything, anything gets into the retina, it will create a response. Actually, it's not because it's a connected to central nervous system. A small anything will create a little bit of a response, but you have conjugated with a drug which is going to inhibit that. Okay, but it is not that it will not create a big immunological response at all. It's a biocompatible.
Thank you, sir. And I have a next question from Skantesh. His question is, in view of nanomedicine, what is your perspective on Ayurveda or Siddha herbo-mineral combinations?
I have not worked in this, but it is a very good area as far as I know for this, whatever this. Because a lot of the already formulations are there, it is also working, but how it works we really do not know. That's what which I understood, especially some of the Siddha medicines. One of my colleagues will show he worked on the nanoparticles were there in that, but how it works exactly people are not understood. But yeah, as now Ayush is giving a lot of importance for making it like how the Western medicine develops, like a cell line to animal model to finally to the phase 1, phase 2, phase 3 trial. But Ayurveda do not believe in this. That's what which I was talking with one of the Ayurveda persons. And he says, human beings are different from the animals. It may work or it may not work. That is true, but that's how the Western medicine are developed. And some of the things are really fascinating. See, the lead is used. Even I have taken lead is used in the formulation. Lead nanoparticle, which is used in the formulation of Ayurveda, and mercury is used in the formulation of Ayurveda. But it's a non-toxic. And then when you analyze it, the mercury is there, and then mercury is neurotoxic. That's what they say. The small amount of mercury is also causes a lot of problem. That's what. But in the formulation, I think one of the papers which I did, I think one Dr. Bellare of IIT Bombay has done a lot of work in Ayurvedic formulations. The mercury, why it is it's a density is very high, 13.6 compared to water. But when the formulated mercury, when you put that, it will not settle down. How they have formulated that, what the mercury, what they have modified the mercury, that there is a process of modifying the mercury. All these things, people have done it in thousands of years back, and it is so fascinating. Now we are trying to understand how they have done it. And if you say the same thing to the Western people, they do not believe in it. So it's a fascinating field, actually, of understanding Ayurveda, and lot of scope, according to me.
Yes. Thank you, sir. I think the question is very well answered. And I have a next question. In your opinion, what are the challenges in the field of nanomedicine, especially from a synthetic point of view?
Getting the consistently, say I might be doing the same thing, but the scaling up is the problem. Scaling up is the major problem in a nanomedicine. In the sense, say suppose I make it 30 milligram or 100 milligram of a formulation, and then you want to scale up to 10 grams or say 1 kg, then say nanoparticle, what is the range of nanoparticles you're producing? And the next thing is, say I told this is a formulation. How this formulation is, it's a suspension, right? Suspension in a liquid we have to give, and the sterile liquid and nanoparticle exactly, and it should be in a suspended form. And then educate the people how to administer nanoparticle. So because most of the people are compounders, are the people who take ordinary people. Every time you cannot go to the doctor to take the drug. So administering the drug. So that's why whatever I have shown is the cancer, and then eye. At least you could go every 2 months or 3 months, and then nanoparticles are injected. In this, the challenge is getting a consistency, batch to batch consistency. That is a very challenging task. Getting the exact consistency of the particle size and then other things. That's why I think it's a eye nanoparticle. Though a lot of research is going on, none of the products are there. I do not know why. None of the products are there.
Yeah.
Because once it gets into then standardization, exactly when you scale up, say 1 kg I am producing today in this batch, the nanoparticle of size is 200 to 250, the range it should be. So producing that in the scale up is the problem. You cannot make it a 30 milligram or 50 milligram at a time and create. Then the cost will be so high. So we are doing it in the lab for the experimentation and for the research. So there should be proper with exactly this nanoparticle size should be produced like that. That is the problem which I am feeling. And then administering that with the common people. That's why the oral medicines are very useful, because anybody can take the medicine.
Hello.
Make sense what I spoke?
Sure, sir. Yeah. My next question is, cancer stem cells have drug resistance property. Can nanoparticles overcome this problem?
Yes, the next thing is that's what which I showed in one of the slides. It is taken up with a different mechanism. Probably it is overtaken. The further work should be done, but some of the work has already been done. Some of the cancer stem cells overexpresses certain receptors. You just make a ligand to that overexpressing receptors, and then it is taken up by the receptor mediated endocytosis. Then the chances of killing cancer cells is high. That's what some of the results have shown. And actually, one of the blood cancer, the cure is there. What they have made is an antibody, which goes and exactly binds, because it's a blood, exactly goes and binds to the cancer antibody, and then it releases there, and the killing takes place. So a lot of research is there, but no product yet. The product is not there. One except the blood cancer. Make sense what I spoke?
Very much, very much.
Yeah. Okay.
Hello. Yes, sir.
Yeah.
Thank you very much for a detailed answer. And through this, we got to know the real challenge in an medicine is associated with scale up and productization. And that is where exactly really we all have to look into. And I would like to pass on to Dr. Damle, sir.
Yeah, I just typed a question there. Are other questions over, Bharat? Are there any questions from outsiders?
Yes, sir. I would like to take another question.
Yeah,
Can you,
finish
sir?
that. Finish that. I will ask my question in the last.
Yeah. Sir, can you talk a bit on gold and silver in nanomedicine?
Oh, I'm
Because
sorry,
in
I've not worked.
Ayurveda,
Yes.
because in Ayurveda, you might have heard of Swarna Prashna and all.
Yes.
So, as we have majority of Ayurvedic audience, it will be really insightful if you talk on this.
Actually, then all of you must be knowing much better than me because I've not worked in this area. I'm sorry, I do not want to speak in shallow way. Yes, as you both silver and both are biocompatible, and then in the body, Ayurveda, a lot of formulations are used with this, but I have not worked in this. Except in they make a sulfur bond with the gold nanoparticle, and then they have done a lot of which I have not done it, and then a lot of people have worked on see where it is going nanoparticle, because one of the important thing is, as I said, the gold nanoparticle, the amount, what I say, the particle size can be actually can be maintained. A 50 nanometer means a 50 nanometer gold nanoparticle can be made exactly. That's why it will be much useful. But not with the whatever we make in a biomaterials. Only the tracking that where it is going, and all people have done a lot of work on this, but I have not done any work, and I do not know much about it. I'm sorry about it.
Yeah, thank you very much. I think one of the things which is coming forward is people of who have studied science and traditional medicine, maybe
Medicine,
orientation,
yes, yeah,
are coming together. Secondly, non-anglicized heart is very important for us to be together. And
yeah, correct.
and I can see that with the multiple talks that we had here, people are open for collaboration, and I think it's very important all the everybody to realize this, including AMAI. Any of your students, your people, PhD graduates want to collaborate. People like Rajguru are open for collaboration, and probably.
Oh, I will be very happy, sir. Actually, in fact, I believe more in Ayurvedic medicine. I mean, I cannot say like for infectious disease you take, but all the other things, especially chronic diseases, Ayurveda works perfect actually. So I believe in that, and then only thing is the validation, and somehow we have to identify how to validate each of this. I meet regularly. You must be knowing Dr. Veliathan. He is in Manipal. He has written a book, and I think he is now in charge of a DBT Ayurveda uh, program. Uh, so he is also, though he was our previous vice chancellor. After his retirement, he got fascinated with Ayurveda and written a book modified the Charaka Samhita into English, and then he understands a lot of Ayurveda and so many things. Say each individuals are same. We cannot give. Now you just look at the personalized medicine is coming. How it is coming, say, we look at the how long time back, thousands of years back, people have identified individuals are not same. Each human being is not same. It depends on vata, pitta, and kapha. Somebody is, it's not, vata person, pitta person, and this. Now you just look at that for the cancer, they're doing the same thing now. The personalized medicine is coming. So that's what some of the diseases, with the chronic diseases especially, they give everybody, they treat the same and give the same dose. It creates a lot of havoc in the society. Then the Western medicine has not understood anything about the body actually, other than they have understood in the one-dimensional way. But Ayurveda understanding is much different way actually. It gives a newer dimension of understanding. So I think I was talking with Dr. Veliathan in one of the drugs which is used for arthritis, like which is a deadly drug, and then it's a once in a week you take. It changes a lot of things. Its side effects are so high. They administer for everybody. Then he started doing some of the things. It is more effective in few of the people where because vata or pitta with some of them, with the certain constitution of the body, this drug is more effective compared to the other people. This is what the identified he was talking about. This new dimension, Indian, I feel the Western medicine, forget about the Western medicine, that the ICMR should identify these things and then should also teach them, whatever the MBBS doctors, some aspect of Indian medicine. In the process, they can understand properly. Say, now I will tell you a simple example. The food what we eat in Western medicine, they do not care. They think about only protein, carbohydrate, and other things. Say, if you go for with a fever, I asked one of the person with the MD, can I take this food? Yeah, you can take any food. I mean, when I was suffering for something, can I have pizza? I asked him. Yeah, you can take pizza also. I mean, any food. Assume somebody with an MD who is going to become a doctor, he cannot understand exactly. We, from past thousands and thousands of 500 years, we have understood what food is so important. Before, people used to identify this food you do not take. Nowadays, people have left even that also, because there is a wide gap between Western medicine, allopathy, people who administer allopathy, and then the people who look into Ayurveda. But Ayurveda says what you eat is medicine. The food you eat like a medicine. The food is very important part of your day-to-day activity. That is people have looked into the health instead of aping Western civilization. We will have to look into what we have in our tradition and adopt. I feel that will be a greater thing. I do not know whatever from my heart I have told.
I think we should collaborate, because truth may be somewhere in between.
Yes, sir. I agree. What is in my heart, I spoke. Actually, I do not know.
Yeah, no, I agree, totally agree, and endorse what you have said. Truth could be somewhere in between, because traditions speak a lot of things, and some of the contradictory things, and science also sometimes speaks contradictory things.
Yes,
so anyway, it is very nice to hear that people are willing to coordinate, collaborate, and hopefully we'll do some work together one day.
Yes, I will love to work.
Sure. And also with our friends in Kerala, because they are really filled with enthusiasm and a lot of resources. Now I request Dr. Shiban Ganju to deliver a vote of thanks.
On behalf of Atrimed Pharma and all the members of the panel, I thank the speaker and all the participants who are far away from us. It was a wonderful session. hopefully we'll have more contact with you and more sessions within future, so we can understand your and others' work much more than what we know now. It was a good initiation to nanomedicine, and hopefully next time you will deliver more fascinating story to us. Thank you, everybody, and we'll see you in the next lecture. Keep on the lookout for our information for the next hour with the renowned scientist. And with that, we'll call it a day. Since nobody has got any questions, we come to close now. Thank you very much.
Thank you very much, sir. Good night.
Thank you. Thank you for giving me an opportunity. I had a wonderful time. Thank you very much.
Thank you. Thank you very much. Thanks, everyone.
Hello.
Hello. Yeah.
Hello.
Yeah.
Yeah. Thank you, sir. Thank you very much.
Yeah. Thank you, Bharat. Thank you. I had a wonderful time. So we'll be in touch.
Thank you, sir. Thank you very much.
Yeah.
Sure, sir. Sure. Thank you.